Howdy, howdy. This is Mr. Justice. We are going to talk about topic 2.4, ecological tolerance. Take a look at the objectives and the essential knowledge. We'll talk a little bit about how one can identify authors' claims today. So keep that in the back of your brain throughout today's lecture. Let's talk a little bit about coral. Coral are these wonderfully biodiverse aquatic ecosystems that are found within shallow waters, right, in marine ecosystems. Coral is a really interesting composite organism in that it is made up of actually two different species of organisms. One, you see here, the kind of lighter color, is the coral itself, and then within the coral, living within the coral, are these photosynthetic algae. And that photosynthetic algae produces sugars for itself, but also for the coral. And the coral, in turn, will give shelter to those algae. It's a tenuous relationship between these two, and it's really impacted by temperature changes. Even just small temperature changes could cause healthy coral to eject that algae and bleach. It's called coral bleaching because that white exoskeleton that is left behind is calcium carbonate rock. It is no longer alive. The coral died, algae is gone. All that's left behind is basically the husk of its former self. And it won't return unless new coral polyps establish themselves on these surfaces. So this is what coral bleaching looks like on the right. Normal coral on the left. An slight increase in temperature even can really impact that coral, ejecting that algae and bleaching the coral white. So I wanted you guys to watch this. So if you're watching this as the lecture, which you are, I'd like you to push pause on my video and then open up a new tab. Go to crjust.us/OFU. It's an island in American Samoa. And you'll watch this video. It's about 3 minutes long. In the video, you'll see coral bleaching. You'll actually see an animation of how it works even at the cellular level. So I'm not going to play it here because this is a video lecture, but you can access this video through crjust.us/OFU. If you watch the video, you'd be able to answer these three questions. The first one, what type of species interaction exists between coral and the algae? The answer would be it's a symbiotic relationship, and this is a great example of mutualism, right? Because the food is provided by the algae and the protection provided by the coral. Number two, why is the photosynthetic symbiont ejected? An increased rate of photosynthesis because of the temperature changes increases the amount of oxygen inside of the coral, and as oxygen molecules in enough concentration can wreak havoc on the interior machinery of the cell. And so in an effort to save itself, the coral cells will eject the algae away. But doing so, it is releasing its food source, and death will occur shortly after. And that's pretty much what coral bleaching is. Coral bleaching is this process where we remove the coral and the algae, living cells. All that will be left behind is the calcium carbonate coral. That's coral bleaching. So we can do experiments, coral experiments, and some experiments were done in Ofu. You see a picture of the island right here. What's interesting about Ofu is that the lagoons on one side of the island have different temperatures than the lagoons on the other side. And so think about it. We've got a warmer side, we've got a cooler side. We'll be able to put some coral from each of those sides, and maybe we can perform like a heat stress process to it to see if it does in fact bleach, right? So here's our research question, right? If we were to do this, do the coral from the warm side of the island have a broader heat tolerance than the coral from the cool pools? That's a research question. We'd have to collect some data to do it. So, right, and come up with an experiment to do it. So here's the experiment. I took, the researchers from the video that I got this from, took the coral from the warm ocean and put it into a stress tank, a heat stress tank. And what it does is it kind of mimics the tidals, the high tide and low tide movement of water, and increases temperatures, decreases temperatures throughout, right? And what they did too is they took some coral from the cool side of the island and heat stressed that separately, right? So we've got two different species of coral, one from that's uniquely adapted to live within the warm side of the island, and then another type of coral that is uniquely adapted to live on the cooler side, right? And we've got some data. So after the heat stress process, researchers found that there were 20% coral bleached that come from the warm pool after the heat stress process, and from the cool pool, 55% of those coral bleached, right? So we got some evidence. Now, if we were to make some claims, what we can do is oftentimes start with our evidence first and allow that evidence to help us write out our claims. Claims are answers to the research question. So in order to answer that research question at the top of our screen, we first have to kind of think about it a little bit, right? Organize our thoughts in the evidence statement first. So let's do that. Let's think to ourselves for a moment. So our evidence here is coral from the warm pool had blank bleaching coral than coral from the cool pool. The answer would be less. We saw that right here, right? 20% versus 55%. Now, based off of that evidence, we're able to make a claim. That, again, that claim is going to answer that research question, right? Here's my claim. Coral from the warm pool are blank able to tolerate high temperatures than coral from the cool pool. What would we put in there? More or less? Well, based off of the evidence, we would suggest that coral from the warm pool are more able to tolerate high temperatures. And I know that because less of that warm coral bleached compared to the cool coral from the cool pool. This is hard to say, right? Cool pool coral had 55% bleaching. All right, we're going to move on here. So when we write out claims and evidence, the hard part sometimes is for us to include those scientific principles. The reasoning statement is just as important. And when we read scientific literature, oftentimes we can look for that evidence and that reasoning to help us try to identify the author's claim. So think about what we would have to do to write a reasoning statement for the evidence and the claim that we wrote on the previous slide, right? We're going to want to explain why coral from the warm pool had greater ecological tolerance compared to the coral from the cool pool. Think about the reason why. We learned it in 2.1 of our topics here together, right? Yeah, it's because of the genetic diversity that exists within the coral species, right? Therefore, some individuals are more heat tolerant, right? They've got these heat tolerant genes that allow them to survive in warmer temperatures. Those coral from the warm pool have that ability, that trait, right? That trait comes specifically from their DNA, their genes. The genes impart their ability to either withstand or not withstand increases of temperature. We see distribution of coral species throughout our world here, generally around the equator, because that's going to be where it's warmest. And as we move away from the equator, from the tropics to the subtropics, 30 north and 30 south, we still see some different species of corals. But notice that we tend to not see them outside of this band of warmth. That's because all coral generally needs to have warmer temperatures within the oceans, 20 to 30 degrees Celsius or so, to live and thrive and survive and reproduce, and really to live and exist within their optimum range. So that brings us to this ecological tolerance curve that you're looking on the screen here, right? The center part, so this is a nice bell-shaped curve, right? So on the x-axis, we'd have some abiotic condition. Could be pH, could be temperature. And on the y-axis, we've got the number of organisms, right? Or a percentage or abundance of organisms within the ecosystem here. And so depending on that abiotic condition, there's going to be an optimum range that any species would be able to exist within that pH or that temperature or whatever the condition is. If we move outside of that optimum range, right? Think about this, that optimum range is determined by the genes of those species. So it's going to be different for every organism, every species, for temperature or pH in this case. So if we move outside of that optimum range, we move into this band here, this zone of physiological stress. And what this area is the species aren't dying necessarily. It's stress, right? Maybe a little heat stress decreases its ability to survive, reproduce, and live to its biotic potential, right? We're increasing the temperature so much that maybe we're damaging the integrities of the cells, or we're decreasing the temperature so much that we're not allowing metabolic processes to continue at the normal rate that they normally would, right? So we've got the optimum range, we've got the tolerance range that makes up the middle and the ends of our bell-shaped curve. And then on the extremes here, on the left and the right, is the zone of intolerance. The zone of intolerance is where death would occur for the species. It's either too cold or too hot for the species to continue to live, depending on what that abiotic factor is. And I mentioned like that abiotic factor, right? What are some of the other ones here? This Goldilocks curve is what we can call it. It's a universal concept. And so it can be applied to other abiotic conditions like salt levels or salinity, right? You see pH here as the x-axis on this example here, but we could also have dissolved oxygen present there too. Temperature, even flow rate, right? Flow rate is referring to the speed of water through a river. If the speed of the water through a river is too high for some rooting plant to take root, right, it will not be able to survive and reproduce and continue to live and fulfill its niche in this ecosystem. So here's what a tolerance curve might look like for coral. This is a species of coral found within Hawaiian waters. Hawaiian waters, ocean temperatures tend to be around 26 degrees, 25 degrees Celsius. So let's take a look at the calcification rate. That's going to be the line with the white circles here. The calcification rate, this is the rate at which calcium carbonate is produced by the coral polyps themselves. And it looks like the optimum range is going to be probably around 25 degrees or so, and then to the left and to the right of that. So maybe a range of about 22 to 26. Below 20 or above 28 is going to be in that zone of intolerance. Organisms wouldn't be able to continue to survive and reproduce at that. But we might see some stress happening here in these colder temperatures or slightly more extreme temperatures over here, right? That coral might not be able to be living and thriving and reproducing at those kind of ends over here. We're going to see the most calcification happening within its optimum range. The other line, just shifted to the right a little bit, is recruitment rate, is what they call it if you're a coral researcher. This is how well the coral larvae are able to establish themselves onto new surfaces. Similar ranges here, right? Somewhere around 26 degrees. And it makes sense because the coral in Hawaii is uniquely adapted to be able to live within 26 degrees or so in the ocean temperatures. So you might think to yourself, why might some individuals in the population be able to withstand increases or decreases in temperature? You got to remember, right? It's all about their genes. The reasoning here. This is our reasoning statement. Their genes impart this ability, right? It gives them a trait to be able to withstand higher temperatures or not, right? Hey, and remember this. Genetically diverse population is going to be better able to respond to environmental stressors. So imagine for a moment maybe an oil spill decreased the amount of coral in this area, and then a year later, right? So we decreased the genetic diversity quite a bit just by killing off a lot of the coral. Then about 10 years later, there we start to see an uptick in ocean temperatures. Because we've already decreased the genetic diversity, we've decreased the number of individuals that would be able to withstand those higher temperatures. So, right, if we decrease those individuals, we decrease the ability of this colony to be able to rebound and bounce back from that stressor. So let's talk a little bit about dissolved oxygen. This is an important abiotic factor here within aquatic ecosystems. Fish are going to absorb dissolved oxygen directly into their gills, right? And different species of fish, based off of their traits, their genes, have different ecological tolerances for dissolved oxygen. So measuring dissolved oxygen is a really important water quality test in determining if a habitat is suitable for different fish species. So let's take a look what these ecological tolerances would look like for these 4 different species of fish. Take a look, right? Hey, which species has the broadest ecological tolerance? Which one has the narrowest? It's going to be the one that is taking up the most space on our x-axis. You can also find it over here too. Look, 14 minus 7 milligrams per liter is about 7 milligrams per liter of tolerance range for trout. So we're going to be able to have a pretty wide base down here, right? Which one has the narrowest? It's going to be pike is 5 to 2. 5 minus 2 is about 3 milligrams per liter of difference on either side of those. Pretty wild stuff, right? It'd be interesting to, let's see, the broadest is the trout, the narrowest is the pike. All right, take a look at this, right? Now imagine that the DO level was 1, right? If it was 1, all of the fish to the right of 1 are going to perish. We've got a hypoxic environment, right? One that has very low oxygen levels. Fish need oxygen to survive. And if we're at 1 doesn't fall within any of the optimum ranges for any of these 4 fish species. But if it was 3, take a look at this, right? If the DO level, the dissolved oxygen level in a lake was 3, that falls within the optimum range of pike, right? This green line over here, but not within the optimum range of any of the other 3 species. That brings us to our question over here. Which species will be most affected if dissolved oxygen concentrations fall below 7 milligrams per liter? So here we are, right? Hey, look at this. Think to yourself, okay, we're at 7. That's more than enough oxygen for pike. 7, we're well within the range of what carp are able to take in, right? We're at 7, so we're more than, well, probably a little bit less than what would be optimum for bluegill, but we're still within that range. So just through the process of elimination, we're able to see that trout will be most affected. And it kind of makes sense, right? Hey, trout need a lot more dissolved oxygen in the water and able to survive and reproduce the best. Hey, explain why trout will be most affected by lower dissolved oxygen. Hey, trout have a low tolerance for low dissolved oxygen levels and therefore cannot thrive and reproduce when dissolved levels fall too low. It's all about their genes. Their traits imparted that ability for this species to be able to not withstand low dissolved oxygen levels, right? And therefore they won't be able to live to their biotic potential. So how can you get dissolved oxygen into water? So you have this picture here because you can kind of see the water flowing over these rocks. Fast-moving water, moving over rocks, tumbling over rocks, heading down waterfalls, incorporates oxygen levels, oxygen back into water, increasing those levels too. Colder water is able to hold on to dissolved gases a little bit more readily, so we'll probably find them within colder lakes as well. Take a look at this. So we're trying to identify the author's claim. So when you go through this and read this paragraph, I want you to think to yourself for a moment, what's the reasoning? What's the evidence? And you can kind of reverse engineer it a little bit, right? Thinking backwards to get to that claim. So here, I'll read this with you. So this is dissolved oxygen concentration of Midwest rivers. It's a concern for brown trout. The loss of suitable brown trout habitat in Midwestern rivers is a concern for conservationists. Trout serve an important ecological role in the biodiversity of river habitats, providing food for bears and eagles. A critical factor in determining trout survival is the level of dissolved oxygen in the water. Researchers conducted a study of 10 rivers throughout the Midwest and measured the concentration of dissolved oxygen in milligrams per liter. Although brown trout were found in waters ranging from 2.6 to 8.6 milligrams per liter, trout in the developmental stage were found only in waters with concentrations of dissolved oxygen above 4.9 milligrams per liter. In addition, levels of dissolved oxygen below 4.9 milligrams per liter were associated with decreased weight in adult brown trout. Which of the following statements best identify the author's claim? Take a moment. Read through, push pause. I'm going to tell you the answer now. The answer is B. And take a look. The reason why is because this is our evidence right here, right? Although brown trout were found in waters ranging from 2.6 milligrams per liter to 8.6, trout in the developmental stage were found only in waters with concentration of dissolved oxygen above 4.9 milligrams per liter. So think about this for a moment, right? Trout in that developmental stage need to be within its optimum range in order to be able to survive. Sure, it can. The trout can survive. The species can survive in 2.6, but in order to be able to grow properly, we've got to have at least 4.9, right? In addition, levels of dissolved oxygen below 4.9 milligrams per liter were associated with decreased weight in adult brown trout. Think about this. That adult brown trout is not living within its optimum range. I know that because the weight is decreased below that, right? So the only one that really works and fits into this then has got to be the answer B. All of the other ones don't necessarily work out. I hope this was helpful. Go ahead and finish up the assignments associated with this day.